Good evening, everyone, and welcome to the Brain Products webinar on stationary EEG and eye tracking. We are really happy to see such a great turnout of attendees tuning in from wherever you are in the world to learn more about our hardware and software solutions for EEG and eye tracking. My name is Christina, and I'm a product manager at Brain Products, and I'll be moderating this event today. I'm very pleased to announce that we'll have presentations by two speakers, Laura Loix and Michael Hofstetter, who I will introduce when I outline the proceedings of the webinar. Our first talk will be regarding the hardware setup for combined EEG and eye tracking studies, and the second one concerns handling the data after you've recorded it. Lastly, we'll follow up with a Q&A. Our first presenter is Dr. Laura Loix. Laura is a scientific consultant in our sales team, and her scientific background is multidisciplinary, drawing on experience in both eye tracking and EEG. Today, she will guide us through setting up with your stationary EEG and eye tracking experiment. She also wrote a very comprehensive guide to getting started with such experiments using Brain Products and Tobii Pro equipment. This was published in our latest press release, and we'll include a link to that in our follow-up email. Since we're going to be doing a live EEG and eye tracking demo today, we have Dr. Eduardo Bellomo and Dr. Saba Gizemilio helping Laura with this procedure. Our second presenter, Michael Hofstetter, is the team lead of our scientific support department, and he has worked extensively with BrainVision Analyzer and comes with a wealth of knowledge for helping guide you through your data analysis procedures. During his presentation, he will walk you through the new Add Channels transform. As you can see, we have some great talks lined up for today. So without further ado, I will hand the presentation over to Laura, who will surely deliver a fantastic talk and a very informative demo.
Thanks for the kind introduction, Christina. Again, these are the topics we want to cover today. So first of all, I will talk about EEG and eye tracking in general a little bit, and then I will show you the hardware that we're going to use today. And I will do a short demo recording with my colleagues Eduardo and Sara. In the end, we'll turn to analysis and also show you how to combine EEG and eye tracking data together. Of course, we cannot show you all possible setups for EEG and eye tracking, so we had to decide for one specific setup for today. And in this context, we're very proud to mention again that we have started a new cooperation with Tobii Pro. So our people have been working with their experts to make this integration of EEG and eye tracking as smooth and as easy as possible. So today I will show you a setup with our EEG Brain Products hardware and with the Tobii eye tracking hardware. All right. Now let's start with some general thoughts on EEG and eye tracking. So what we are doing here is combining two very different sources of information. With EEG, we want to monitor brain activity, so neural activity basically. And with eye tracking, we can gain two different kinds of information. One is the gaze position, so where somebody is looking at, and the second one is the pupil size. Now, obviously, there are many reasons why someone want to combine EEG and eye tracking, but I would like to just talk about a few examples. First, the pupil, for example, responds with dilation to different stimuli, for example, to emotional arousal or to cognitive effort. So one might just want to add pupil size as a very sensitive physiological measure to the EEG experiment and gain additional information this way. A second thing is that, of course, eye tracking tells us about the gaze position, and this tells us where the open focus of attention is of our participant. So what we can do is we can check if the participant is really focusing on the task, really focusing on the targets we're presenting, or if they may be looking elsewhere and not paying attention. With EEG alone, we might not be able to know this, but with eye tracking, we can gain this information, check if the participant is actually focused on the screen where they should, and then use this to correct our EEG analysis and, for example, discard trials where the participant wasn't paying attention. And the third example I have is that eye tracking gives us very precise timing information about when some point on the screen might be fixated. So for EEG research, it's often enough to just time lock the brain activity to the presentation of a stimulus, and you would just rely that the participant will focus on the stimulus and then calculate event-related potentials. But for some research questions, this might not be enough. So you might want to know when precisely the target is found in a visual search task. So you need to know this precise fixation timing on the target. Or for example, in a reading task, you might want to know when exactly is is a certain word fixated with the eyes. Of course, we cannot know this information with EEG alone, but we would need simultaneous eye tracking to gain this information and then use it for our EEG analysis. And then we can, for example, calculate fixation-related potentials. All right, so in each of these examples, the eye tracking has really valuable information to offer for our EEG analysis. Now, if we want to use EEG and eye tracking together, we have to bring them somehow onto the same timeline so we can use this timing information from the eye tracking for the EEG analysis. Now, how do we do this? So first of all, what we will need are some kind of synchronization events. And they have to appear at the exact same time point in both data sets, in both the EEG and the eye tracking data, so we know which time points correspond. Why do we need these synchronization events? So just to keep this in mind, EEG and eye tracking are very independent data sets. So they are recorded by different devices with different software, maybe at different sampling rates. And this way, if you have these synchronization markers in both data sets, you can at least tell which time points should correspond to each other. If you have those, you can stretch one of the data sets to match the timeline of the other. In most cases, you will try and upsample or stretch the eye tracking data to match the EEG data that is usually sampled at a higher rate. And then in the end, once they're on the same timeline, you can use any kinds of events you have detected in your eye tracking data and import them to your EEG data for EEG analysis. So this is what we want to do today. Luckily, the last two steps, so the stretching of the eye tracking data and the importing of the eye tracking events to your EEG data, this will be done by BrainVision Analyzer, and you don't really have to take care of this at all. This will happen automatically. What we need to take care of and what you should remember for later is that we need to set up these synchronization events in both data sets. All right. So today we will walk you through the whole process, how you could pick your hardware, set up an experiment, and perform these recordings and analysis. So I will stick to that workflow on the slides. First of all, I'll talk about the hardware we're going to use for today. Then I will talk about the experiment we are setting up, and also how you can set up these shared synchronization events in EEG and eye tracking. And then we'll perform a very short demo recording. So this is just to illustrate how the recording works. And in the end, we will analyze a larger pre-recorded data set that will show us how to look at the eye tracking data and how to merge the eye tracking data with the EEG data. Okay, so let me start with the hardware. First of all, with the eye tracking hardware. So today we're going to use the Spectrum. The Spectrum is over here on my left side. This is Tobii's most powerful screen-based eye tracker. So this is made for stationary eye tracking. So the participant is seated in front of the eye tracking unit down here and looking in the direction of the eye tracker, basically above it at the screen, or yeah, whatever you are presenting up here. The Spectrum comes with a pre-mounted monitor that you can see here, but it can also be used with an external monitor, or it can be used for a defined section of a room. So this makes it quite optimal for eye tracking with, for example, children who might not pay attention to a screen and who might rather pay attention to a small scenario you're building behind the eye tracker. The Spectrum can perform really reliable eye tracking also while giving some freedom of head movement. So it can recognize the eyes in a certain range, so a certain operating distance from the tracker, and allows for some freedom of head movement. So it doesn't require a fixation of the head or a headrest or something like this, but the eye tracking can be really natural by just sitting in front of the eye tracker. Again, this is great for naturalistic viewing and also for populations like small children who might not sit entirely still in front of the eye tracker. The sampling rate of the Spectrum is quite high, so it goes up to 1,200 Hertz. And with this sampling rate, you can detect even the quickest eye movements like microsaccades. It also has a very high accuracy and precision, and the Spectrum performs binocular eye tracking. That means that both eyes are being captured by stereo images, and each eye can be tracked separately. So the gaze position, the pupil diameter, and the pupil position in space will be tracked for each eye. The Spectrum can also receive hardware triggers. So here at the back, it has an 8-bit input port for TTL triggers, and it connects to the recording laptop, which is standing over here, via Ethernet cable. The calibration and recording with the Spectrum can be performed most easily with the Software Pro Lab, and I will tell you a lot more about this software later on. All right, so let's take a look at our EEG setup. The electrode system that we want to use today is the RNet here beside me. So it could also be a different cap, just note we are choosing this for simplicity today. So in the RNet, the electrodes are integrated into a very light and comfortable silicone net under these small dots here, and they are covered by small sponges. So basically the electrodes make contact with the scalp via small sponges that are soaked in a saline solution. And they are located in the positions of the 10-20 standard layout. The core feature of the RNet is that it's extremely easy and fast to put on, and it's very comfortable for the participant with really minimal effort. So the whole net just has to be soaked in a saline solution for 10 minutes. It's placed on the head, and you're basically ready to go. So you don't need to abrade the skin, you don't need to use any gel, and you won't have any gel residuals in the participant's hair. So again, this is perfect for populations like small children or infants, or also for clinical populations. Because of the gel-free technology, the optimal impedance levels of the RNet lie between 60 and 100 kilo ohm. And this technology is perfect for ERP research. So this is going to be perfect for our purposes today. You could also perform frequency analysis of up to 100 Hertz. And you can record with the RNet for about 1 to 1.5 hours before the sponges start drying out, and you would need to add a bit more water. All right, for amplifying the EEG signal from the RNet, we're going to use the actiCHamp Plus. So this is our most versatile lab amplifier. And the electrode inputs come on top in 32 channel modules. So you can put in up to 5 32 channel modules. This one has just one, but you could potentially measure from up to 160 channels. The actiCHamp Plus is our improvement of the actiCHamp, and it can use all kinds of electrode technology, so it can work with the passive RNet, but it could also work with actiCHamp electrodes, which are of active technology. The sampling rate of this amplifier can go really high, up to 100,000 Hertz, which is quite special about this amplifier. A very nice feature are also these auxiliary channels that you can see in front. So there are 8 auxiliary channels to which, from which you can record data with additional sensors. So for example, these could be physiological sensors, or it could be a photo sensor or something like this. The actiCHamp Plus also has, the actiCHamp Plus also has 2 8-bit trigger ports at the back. This is what they look like. I don't think you can see it like this. One of the ports is a trigger input port. So here you can take in up to 255 different trigger signals. And the other one is a trigger output port. And there you could just, if you wanted to, you could just mirror the trigger signals that come in and pass them on to another device. And lastly, here underneath the amplifier, we see the battery that is used. This is the power unit. This is lithium-ion based and has a very constant and reliable discharge. And when it's fully charged, you can record up to 24 hours with it. I want to show you 2 more pieces of hardware for our EEG setup that we're going to use today. The first one is the photo sensor. This is a very tiny sensor. I can also show it on the screen. And it can detect changes in brightness. So if we attach this to a screen, for example, then it can continuously track the changes in screen brightness and is being put out here in this auxiliary cable. And the second device we want to use is the StimTrack. This is the StimTrack. It's just a tiny box, and it can take in signal from sensors like the photo sensor that can just plug in here. And what the StimTrack does is it transfers this continuous sensor signal into trigger pulses. So if the sensor signal surpasses a certain threshold that we set, it sends out trigger pulses out of this BNC port on the top, and they can be fed into our amplifier. So this is exactly what we want to do. We want to connect the photosensor to the stim track and then set a certain threshold in the stim track with these screws here. And then whenever the threshold is surpassed, this will send a trigger pulse into our actiCHamp Plus. And at the same time, for completion, we're also going to record the continuous signal from the photosensor in one of these auxiliary channels here in front. Okay, and then finally, to complete our EEG setup, of course, we also need software. For recording, we use BrainVision Recorder, and for the analysis, we'll use BrainVision Analyzer. You will see both of those later, and Analyzer in more detail, because here is also where we're going to merge the eye tracking and the EEG data. Okay, having looked at our hardware setup, let us turn to the experiment, and let's see how we can set up these synchronization events for both the Spectrum and the actiCHamp Plus to have them aligned in both eye tracking and EEG data. For setting up an experiment, at first we have to decide for an experiment control software. And today we're going to use Tobii Pro Lab. So the great thing about this software is that you can use it for the whole process. So for designing experiments and presenting them, then for recording eye tracking data, and also for analyzing it. The experimental design part allows you to present stimuli, so this is comparable to other software like ePrime or presentations like Toolbox, whatever you might be using. And here the nice thing is that setting up timelines and experiments is really easy and intuitive, basically with drag and drop, and also setting up trigger signals is really easy. Then the recording part is also extremely easy with ProLab, obviously. So this is working for all screen-based eye trackers, not just the Spectrum, but for example, also for the Fusion eye tracker. And here you can also record, monitor the recording live and see where on the screen the participant is focusing. And lastly, ProLab has many great analysis tools for eye tracking data. So even really complex analysis like mapping gaze data onto moving stimuli or onto videos is really simple and just takes a few clicks with ProLab. Sadly, we won't have time for going into really fancy eye tracking analysis today, but please take a look at the website from Tobii Pro and take a look at ProLab's analysis features. Okay, so for today, we're using ProLab for all these three things: for designing the experiment, recording eye tracking data, and analyzing it. The task we have prepared is very simple. Namely, it is a checkerboard task. So here the participant just has to focus on the middle of the screen and will see two alternating pictures of a checkerboard that swap every 500 milliseconds. So we have based this task on a paper by Sandman and colleagues. In the EEG data, these checkerboard stimuli should cause a very clear and simple visually evoked potential. So this should be very pronounced in occipital channels, and what I have plotted here is an example for OZ, O1, and O2. And for the eye tracking data, we also want to perform a very simple analysis. Namely, we want to check when and if the participant is actually focusing on the center of the screen. So we are going to define an area of interest, here marked in red on the checkerboard stimulus, and we'll see, we'll identify when the participant is focusing into this area of interest, so when there's a hit, or when they are looking away. And for the combined analysis, we want to use this information from the eye tracking and inform our EEG data with it. So that means we want to reject all trials during which the participant was not focusing in the area of interest, so when they were not paying attention. And our hope or our goal here is to get cleaner EEG data. So by discarding all these invalid trials where the participant didn't pay attention, we hope to get a nicer event-related potential in the end. All right, so as we said before, for a combined analysis, we need to bring these data sets onto the same timeline. So we need synchronization events to identify common points in time in both. So luckily, we're using ProLab for experiment control and also for recording eye tracking data. So all stimuli that we present in ProLab will automatically be also marked in the eye tracking data right away. If we had a computer with a parallel port, we could also set up nice trigger signals in ProLab and pass them right on to our actiCHamp Plus. However, for today's setup, we're assuming you might just have a laptop that doesn't have a parallel port. So we want to show you how we can get these synchronization markers without a parallel port, but with the help of the photo sensor and the stim track. To show you how we do this, I want to show you the task. So this is what it will look like in ProLab. Basically, we always start with a calibration procedure, and then we have a timeline where the main part of the experiment are these checkerboard stimuli that alternate. So as I said, each for 500 milliseconds, and we have set up 100 repetitions each. The task will then need to be framed by instructions, fixation crosses, and whatever not. But we also want synchronization events that happen right before the task and right after the task. So as I said, if we add a sync stimulus in the eye tracking, in the eye tracking task in ProLab, this is also going to be written to the eye tracking data. We don't have to care about that. But how do we get them into the EEG? So what we do is we slightly modify our stimuli, and we insert these tiny bright circles in the corner of one screen, of the screen. And then on top of these white circles, we will attach the photosensor. So what happens is the photosensor picks up the brightness of the screen at this very location. And every time this bright circle is there, it will show a higher step in the signal. And then when it disappears again, you will have a fall in the signal again, rise and fall, and so on. So basically, this is how we detect these two synchronization events at the beginning and the end. And we have also marked every second stimulus with this. And then what we do is we feed the photosensor signal into our stim track, and we apply a threshold. So every time there's a rise in the photosensor signal at the beginning of these marked stimuli, we will receive a marker in our EEG data. In our EEG data, it's going to be called E1. And yeah, basically for aligning the EEG data and the eye tracking data, we will be focusing on these synchronization events at the very start and the very end. So for the EEG data, we will mainly look at the very first E1 marker and the last E1 marker and use those for aligning them to the eye tracking data. All right, so now we have looked at the hardware, we have looked at the experiment in ProLab, and we have made sure we have synchronization events in both data streams. So we can now get ready for our demo recording. So while I'm speaking here, my colleagues have already prepared an RNet on my colleague Eduardo's head, so they're going to come in right now. And we're going to turn on the second camera we have in the room here, so you can see the setup a little bit better. So Eduardo's going to sit down in front of the Spectrum eye tracker and face the eye tracker here and look at that screen. I'm going to hand over the actiCHamp Plus. And we're connecting everything. While we're setting up, just let me remind you, this is a demo recording. It's going to be a bit shorter. It's mainly to show you how the setup works. And we're going to analyze a longer data set later on that has all complete 100 stimulus repetition each. Okay, step track. Perfect. All right. Then let me show my different screen. 1 second. Yes, okay. I'm just gonna stop sharing and start sharing again. Here we go. Okay, you should be able to see BrainVision Recorder on the left side now and Tobii Pro Lab on the right side. So in ProLab, you can see the task that starts with the calibration, then here all kinds of fixation crosses, an EEG synchronization marker, and again a fixation cross. And then in this group here, we have the two stimuli that are gonna alternate. So the checkerboard stimuli, each is gonna be shown a few times here. And then in the end, we have another synchronization event. In BrainVision Recorder, we should be able to see some data now if everything was set up nicely. And perfect, works fine. Okay, so what you can see here are the EEG channels. Did you just? Nice, okay. Okay, so if Eduardo blinks a few times, you're gonna see, yeah, you're gonna see the blinks on the frontal channels. Maybe if you close your eyes, we'll see some alpha here in the occipital channels, as you can see. Yeah, all right, perfect. So our EEG data looks quite fine. And we're basically ready to go. Okay, then let me go from the design tab here in ProLab to the recording tab. The eye tracker is now switching on. And we have already set everything up. So the presentation screen is going to be this one. This is the screen one. And I want to select Eduardo as participant. And then we will start recording. And you can already see that his eyes are being recognized here by the eye tracker. These are these two white dots. If you blink a few times, yeah, they disappear. Okay, now let's start recording. And as you can see here, the spectrum recognizes how far away Eduardo is, exactly where he is in the box. So there's quite a big tolerance for where he could be sitting, and it still captures the eyes. Optimal is something like 65 centimeters distance from the screen. And if you blink again, yeah, you can tell the eyes disappear and reappear. So I think we're fine here. I should be recognized. And I'm gonna start with the calibration. So Eduardo's gonna see only a dot in the middle right now and only a dot in the upper right corner right now. But on this screen, we can follow live how the calibration is working. So these outer 4 points are the calibration points. And then these inner points here are for validating the calibration that we have just performed. Looking really good. Okay. So now here we can see the precision and accuracy that we have reached with our calibration and validation. Here in that point, there was a little bit of trouble, but for our demo recording, we don't really care. We think this is fine. Okay, then I'm gonna continue. And ProLab is gonna start the task now. So we have set up these instructions that at this point, we have to start the EEG recording. So now the screen over here is black. The photo sensor shouldn't pick up anything. So I'm going to start my recording here. Just a simple click here and save. And now here you can tell that the data is being saved in recorder. Okay, so to continue with the task, I'm going to press on enter. We are now ready to start. Please keep your focus on the center of the screen. Okay, and I'll start the checkerboard task. So now I would like you to focus on the bottom right here. You saw this white dot appear. And whenever the white dot appears on the screen here, you see what the photo sensor and the stim track make of it. The stim track battery is actually empty. So in the first trials it worked. So we should maybe exchange the battery and run it again. But you can see the photo sensor signal that rises and falls every time this white dot is present. It rises, and every time it disappears, the signal falls again. So this task is very short. This was the second synchronization marker, and the task is complete. So now I should stop the EEG recording. Let me briefly do that. Okay. Should we try once more with some batteries? I have some replacement batteries here. Okay. These are just simple, normal household batteries. Does it work?
Yeah.
Other way around, from the bottom.
No. Yeah.
We need someone with fingernails here. Thank you. Okay. That way. Exactly. Okay. Let's give it another go.
Great.
Because this task is so short. We can do another one. Okay, I'm gonna save it. Okay. Again, Eduardo is the participant. I'll start the recording, and this time I'm just I'm gonna skip the calibration because we just did it. Eduardo is still sitting in the same place. If you were recording an actual nice experiment, you should always calibrate in the beginning. And okay, so now I start the EEG recording. I'm appending this in this case just for fun. And then we'll start the task again. And you should see the dot here. And now actually the stim track has battery again. So now you can see every time the photosensor detects this white dot, the stim track is writing this E1 marker into our EEG data. So these markers for the stimuli can be used later on if you want to, but you can also just use the synchronization markers. Now, if you look away, maybe Eduardo, if you just exactly, the participant is looking around here, you won't really be able to tell in the EEG data, but from our eye tracking data, we would be able to tell if he was focusing in the middle or not. Okay, thanks a lot. So now I stop the EEG recording again, and we're done with the task. Okay, thanks a lot. Thank you for your time. Okay, I'll stop monitoring recorder here. Okay, so while the others are wrapping up, we stop sharing the camera. Let us jump to Tobii Pro Lab and to the analysis part. So Pro Lab has this Analyze tab here, and I can see all the recordings that I've previously performed in this project file. So what I want to do is now to analyze some pre-recorded data. This is also the data set that my colleague Michael is later on going to use. So it's a bit of a longer data set containing more trials, but it's exactly the same task that we did just now. So you can see here at the bottom is the timeline of the experiment. I can click through here. In the beginning, there are the instructions, and then come the different checkerboard stimuli. If I want to, I can replay what has been presented. Right now, it's a bit confused. Okay. And what you can see in blue down here are the X coordinates of the fixation. So this is basically where on the screen left and right the participant was focusing. And the orange line is the up and down coordinates. So if you're, for example, reading, then you will find this jumping pattern here in the X and the Y coordinates. Okay, so what you can see here, these tiny triangles, these are markers that were written into the data automatically whenever a stimulus was being presented. And the tiny one here in the beginning, these are some instructions. And then this is our EEG sync marker. So we have put this here in the data, and it's being captured automatically. I think something with the video is a bit wrong right now, so I cannot replay it. I would probably have to restart ProLab for this to work properly. Show effect. Okay, let's see if I can show you some of the analysis processes. So ProLab has some really nice visualizations. So if you want to know where on the presented stimulus your subject is focusing at, you might want to create a heat map. So for example, I'm taking the picture of stimulus 1 here, and I can see right away, okay, most of the time the participant was really focusing in the middle. This is indicated by these hot colors. And then some of the time, something is going on in the corners here. Still, in my EEG data, I wouldn't know this. And in the heat map, I maybe also can't tell properly, but I can also look at a gaze plot. So this is now mapping the gaze movements, and you can see they were all over the place. So my participant was mostly focusing on the middle, but sometimes they were looking away into the corners. So what we want to do now is to extract some information from our eye tracking data when the participant was actually focused in the middle, to be able to discard all the other trials where they weren't paying attention. And for this, there is this AOI tool. This is the area of interest tool. And what we can do here is we can draw an area on our stimulus where we are interested in if the subject was focused there. So for example, if I want to know when somebody was focusing on the fixation cross, I could just draw a rectangle here on top of it. But I'm interested in the checkerboard stimuli. So what we did was we prepared these circular areas of interest, so with that circle tool here, and we did that for both checkerboard stimuli at the same time. So every time the participant focuses in here, we get this information that there is an AOI hit, so an area of interest hit. And these events are going to be very valuable for our EEG analysis. So we want to export them now. And for this, I go to the data export. So basically, we have just drawn this area of interest and thereby analyze the data already. And the information about the area of interest hits is already stored here and available for export. So all these options that are ticked here are going to be exported now. So I have information about the recording in general, the participant name, the time and date, and so on. Then, of course, I have data. So for example, the gaze direction at any time. And then I have all the events that were being marked in my eye tracking data. And I want to export all of this in a file now and import it to my EEG data. And what we have to export there is this Tobii Pro Lab output format. So you can also export in TSV or an Excel file if you want to use this with some other software. But if you want to import the eye tracking data into ProLab, you would need into Analyzer, pardon, then you would need to select this Tobii Pro Lab output format. And then just select the recording you want to export here, and then hit the export button. So at this point, I would already like to hand over to my colleague Michael. So we have now analyzed the eye tracking data. We have exported this file from our longer recording. And now Michael is going to show you how to work with this in BrainVision Analyzer.
Okay, so perfect. Thanks for this first part, Laura, for this hardware part. And also thanks to Christina for the kind introduction from the beginning. So I would take it from here, and now we would switch over to BrainVision Analyzer and look into the data and do something out of the combination of EEG and eye tracking. But let me first start with a couple of short introductory words. So again, why are we actually doing this? So the objectives in the second part here is now to give you a guideline to the new pipeline of the channels transformation. And the whole aim of this transformation is to synchronize the EEG and the eye tracking data and to import all needed data and features from the eye tracker into Analyzer 2. The second part here will be to give you also a short outlook to what can be done with the synchronized and imported data. And we will show you this very simple example pipeline. So exactly what you have seen in the demo right now, but as Laura said, a little bit longer recording that had been prepared in advance. Of course, there are also other things you can do, but just for the sake of showing this in a rather short time, we decided for something pretty simple. Okay, let me just give you a couple of words about the prerequisites for channels. First of all, this is an add-on license, so it can be purchased additionally to your BrainVision Analyzer 2 license, and it can be added to either your local or your network dongle. As Laura already mentioned, the prerequisite for the fusion is common markers in both the eye tracking and the EEG data at the same time. We have already seen a little bit about the synchronization. I will also come back to it in the next slide. But then also I want to mention what can we actually synchronize with channels. One thing that we will show today is the Tobii Pro Lab output format from the Tobii Pro Lab and, for example, from the Spectrum eye tracker. However, we can also read some other formats with channels, some Tobii Studio formats, and also some formats from other manufacturers, from SR Research, from SMI, and from ASL. I just want to highlight this pipeline that you will see in Analyzer now, which has been recently added in Analyzer 2.2.1. Is specifically implemented for the Tobii Pro Lab output format right now. So in case you were importing one of the other formats that you see here, it would look a little bit different. However, in the manual of Analyzer, you can actually find the explanations for both of the pipelines. But yeah, what we are looking at is currently just implemented for this PLOF format. Okay. And then just to mention again, the synchronization. This is, of course, the main part of the channels module. It uses the first and last marker method. So that means we need the start and the end marker at the same time in both of the recordings. And the nice thing of having both the start and the end marker is that we can correct for both clock offsets and drifts between the two devices. And then we can actually test afterwards if there are still some residual delays. That there are some delays is actually quite normal, but we can actually check that they are as minimal as possible. And I will also show you how this can be done. And for example, we would use other common events, so like the stimulus markers. One big part of this synchronization is the resampling. So as Laura mentioned, the EEG track data would usually be sampled at a higher frequency, and then the eye tracker would be a bit slower sampled. So we would actually have to resample the eye tracking data. So there is like a match between the two data points from both sources. And this entails some interpolation of the eye tracking data as well. Okay, you have just seen it in the demo, but I just thought I show you once more again the paradigm to remind you. So we have the synchronization events in the beginning and in the end, and then in the middle the 100 trials that take 500 milliseconds each, and then there is always this flickering between the two checkerboard patterns. And what we have here is the photosensor signal below, and we also saw now in the second attempt that the E1 markers were created by the stim track very nicely and consistently, and these will be the markers that we are making use of now also in our demo. Then I just want to also mention the parameters. So 32 channels were recorded with the RNet, and the photosensor is one of those. My colleagues used for this recording a 5 kilohertz sampling rate, and it's also just roughly 2 minutes long, as this is a very fast paradigm. And the data was online referenced to the FZ channel. Okay, and then we are actually in this nice workflow already at the very last step, which is merging the EEG and eye tracking data. And then, of course, we can also do something with the data afterwards. So let me actually switch now to BrainVision Analyzer. And I have already imported the data and opened it. So imported data via adding it to the workspace, which is how we read data in Analyzer. And you see here the data is represented in this primary history window. And we also have here already some processing steps done, and they are represented in this history tree structure, which is how we represent processing steps in Analyzer. And then I have already opened the raw data and it's visualized here. We can see also down here in the timeline that there are markers appearing in the data. So if I click through the data, we see actually here our E1 markers. And if I would just use the photosensor here on the bottom, so this is actually what you see here, because the amplitude is kind of ideal here now in the visualization for the EEG channels. So let me actually double click here and pick out just the photosensor, and then I downscale the amplitude. Then it looks like we saw it in recorder. And now we can see here very nicely these E1 stimuli that come from the stim track and show us every time this stimulus with the white dot in the lower corner. Okay, then I go back to all data, increase again a little bit the amplitude so that the EEG channels are visible. So you see already this might be a little bit annoying having the photosensor on top of this, which is why I also remove it later. But let's first go through the data. So I can just click through the data via the navigation buttons down here. Looks, after all, very nice. Yeah, so the most important part in Analyzer actually here in the menu is the transformations menu, because here you find all the processing steps that you will need. Also the add channels module is here. Down here we also have some visualization options, And yeah, all the data that I will show will be, or that I click on, will be shown here in the visualization window. So in case you're new to Analyzer, just that you can orient yourself a little bit, I wanted to give you this short introduction to it. But now let's actually step into the data. So I have already done some processing here. So there are 3 processing steps applied. The first one is rename the sync markers. If I go to the very beginning, I can see also my first marker, and that's the same is true for the last one. They are called E1 because this is the stimulus that the stim track writes out. I can use it like this, but I thought I just rename them because then it might be easier to identify once I do the synchronization. So if I click here on this edit markers node, which is one of the transformations, I can see now this is called EEG Sync, and this is the same as in the eye tracking data, so I will have it very easy to match the two. The second thing I've done is now actually remove the photosensor, and I did this because we have now created our markers from the photosensor via the stim track, so we don't need it anymore for our example analysis here. So I can actually then just remove it with edit channels transformation, and then I also don't have to pay attention anymore to 2 different types of amplitude ranges that I have in these signals, so that's quite nice. The third thing I have prepared here is a filters node. This is using Analyzer's IIR filters transformation. This is applying a phase shift free Butterworth filter. And the filter coefficients that we have used here, we have applied a band pass filter from 1 to 45 Hertz and also a notch filter at 50 Hertz. These are actually the parameters that we have adopted from the Sandman paper that Laura mentioned earlier, which is the basis of our task today. And if I double click on this node, I can see right away that the data now looks actually very clean. So this is where I would now like to start with, including my eye tracking data. Okay, so there are 2 things I could do actually right now. The first thing is I could simply do ERPs to my E1 stimuli, so to all the checkerboard stimuli, and then I would just create an average out of all my trials, and I would have the classical ERP, and I could look at the visual evoked potential at occipital channels, which is what I would expect when my subject looks at the stimulus. However, I could also make use of the eye tracker because, as we know from the demo, our subject looked away from time to time and did not pay attention to the center of the stimulus, to the area of interest, which means there might be trials in there where we actually don't have a very nice visually evoked potential, and so they might just contribute a lot to the average and our ERP might get much smaller. So it might not really reflect what happens when the subject attends the stimulus. So that's why the second pipeline would be just make use of those trials where we actually can control with the eye tracker that the subject attended our stimulus. And I want to show both types of pipelines to you. And I will actually start with importing the eye tracking data via channels. So to this end, let me now actually close down the nodes, the views of the nodes of the other nodes, so that only that node here with the filters is visible, because that means I will now apply my add channels to this node. And then I call it from the menu. Okay, so let's start with the first part. And this is the file selection. So you can always see on top here what is this dialog meant for. And let me actually increase the size here so that you can see everything very well, because there's some small writing here. So I will now actually add my Tobii POLF file, and I can do this by clicking plus here and then select it from my workspace. So I have actually in my workspace here these 3 folders, my raw files, my history files. This is where the processing steps are saved, and something for my exports. And in my raw file folder, there are my 3 EEG files. And then I also have here this external folder. and there is my PLOF file inside. So I can just open that. That just takes a second. I could actually also drag and drop the file from Windows Explorer into this window here. That would also work. So now it's here, and now we can see right away the path of the file. We can see there are 30 channels inside of the file, and also we see what format it is. So this is recognized from this file right away. Two things I want to mention here. First of all, you can actually add more than one file here if they are of the same format. So if I would have more Tobii PLOF files, I could add more of them. And I could combine them all with the same EEG recording. The second thing is that I see here the file path with some placeholders, and this is quite practical because if I save my PLOF file for all subjects in the same way, then I can actually make use of Analyzer's automation via history templates, and then these placeholders will in every case used to replace the subject names, for example, in the path, and then I can very easily apply this kind of analysis to other subjects. Okay, then let's actually switch to the next dialog page, and this is now the synchronization. So what I can see here on top is my markers in the active node, and on the bottom, the markers in the import file. And since I renamed my markers for synchronization to EEG Sync, I can very easily now take them out here. So I just click select, and now they are here in the selected markers window. I have now to do the same for the eye tracker, and they are, where are they? Here. There are a couple of more markers, of course, but the EEG Sync markers are the ones we need. And what I can do now is I can check, do I actually have two of them in the start and in the end? So let's click on details here. And then I see in this pop-up window, I have a count of 2 for both data sources. And if I look at the first last distance, because the markers should have been sent into both sources at the same time, I can see that this is also pretty much the same distance. So this is already kind of aligned. Then I close this again. And now I have mentioned earlier that I can test for these residuals that would still be there for the latency between the markers of eye tracking and EEG recordings, which is just something that normally happens when you use multiple devices and sync them. And I could test this now. And I would not test this based on the synchronization markers, but I would rather test this on my stimulus markers. So down here where it says timing accuracy test, I will now select based on different test markers, and this will then now add another dialog page. So I click on next, and I have a page timing accuracy test. Otherwise, it looks the same. I have markers in active node and in import files, and I have the same markers, obviously. But this time, I will select my E1 markers, so the stim track created markers in my EEG recording. And now I need to select the corresponding markers from the eye tracker. They are actually not called E1, so they are called StimW. So I select those. StimW because W for white, because this is when the photosensor picked up the very bright white spot on the bottom. So we called it that way to have a very clear stimulus that we can identify here. There's also another one that is just called Stim. That's when the photosensor was picking up the black surface. So that's the other 50 patterns. And what I can do now again is check here first. Do I have the same amount? And actually I do. So no markers were lost, which could happen sometimes maybe. So I have 100 in both of them, actually. So let's close this again. And then I can already make the synchronization test. And this is down here in sync infos. Now I see here some statistics about the synchronization. So I see, for example, the mean synchronization delay in milliseconds. And the nice thing here is I have some delay. It's about 0.8 milliseconds, but it seems that all my markers are kind of falling around this mean. So it's not totally skewed, but it's rather nicely distributed distribution of delays. If I would have now selected the wrong type of markers, so the other stimuli that are not in sync with our E1 markers, I would see this because I would see much larger numbers here, because remember, they are 500 milliseconds apart from each other. So I would just accept this. This is a delay that is way below, well, not way below, but it's below 1 millisecond, so it's very, very short. If I would have now included several PLOF files, so from multiple sources, I could actually now check the synchronization test infos for all of them. So I could now skip from one statistic to the next, but since I only have one file, of course, this is not the case. Okay, then I can actually click next. And now we are at the part where we are not talking about the synchronization anymore, but rather of what do we want to take in from the eye tracker. Yeah. So first of all, we need to select from what device do we want to import. And there are more than one here, which might surprise you. So there's the eye tracker, but there are also events from the mouse and also some common events, so stuff that is sent to both of them. But what we actually want now to include is just eye tracking data. So that's 28 channels, as you can see. So the other 2 channels that we have seen, so 30 channels it said in the beginning, are actually from the mouse. So we ignore those and just select the eye tracker. And that means we can now, in this channel selection field down here, select whatever we want from the eye tracker. And I could, of course, now import everything, but I'm actually not interested in everything. Just for the sake of the demonstration, I will constrain myself to the first 2 ones and select those. And those are the gaze point channels X and Y. So this gives me the X and Y positions of the gaze in pixels on the screen. And I just want to import this a little bit as a proof of principle, because I can look at these gaze point data then together with my frontopolar channels, where I would usually pick up ocular activity in the EEG and can look, does this actually match? Okay, then I select next, and on the next page, which is actually the last one, I can select what markers I want to import. And here again, I could just import everything, but I only want to import one particular marker. Because there are markers for all fixations and saccades, and there are calibration markers. But what I'm actually interested in are the AOI markers, so the area of interest markers that Laura mentioned. And we have a couple of these here, and I would just use the hits. So they're called hits, center checkerboard. Center checkerboard is the name of our AOI. And this is the fixation inside of our area of interest. So this is exactly what I want for this kind of analysis that we are planning, where we want to make sure that really the subject was fixating inside of the area of interest while performing the task. So I will select this again and just take this over. Lastly, I just need to decide as a marker output, do I want intervals or pairs of start and end markers? And I just go for intervals now. And lastly, I can do, I can save my settings into a settings file. This is quite nice if you want also to work with, very regularly with the same type of file, with the same stuff inside, but not just maybe in this analyzer workspace, but also in other workspaces or on other computers, or maybe your colleague is doing another study with the same type of data files. Because then if you say, we always import the same stuff, we always use the same parameters, save it into a file, and then you can reuse it in a different workspace again. So this is again very practical for automation. Now I will actually click finish, and the data is imported, and that should go rather quickly because it's rather short. Okay, and here it is. And we can see right away in our visualization, there are now 2 more channels here, the gaze point X and Y. And I can click now through the data. Well, I should click on the right button. And here I also see my AOI markers. You can also see them down here in the representation of the whole timeline. So these black surfaces here are just a lot of markers in a very close sequence. And this is because our subject has looked for a while into the center of the checkerboard, then for a while away and back into the center. So what we actually have is AOI hits for consecutive stimuli, and then we have misses for consecutive stimuli. Yeah, so what do we want to do now is with the analysis, we want to create an ERP that is just taking care of including all of these trials inside of the blue markers and not of those outside. Yeah, we will just drop those. Okay. Then what I also wanted to show you quickly is the gaze channels together with our frontopolar channels. So I actually mark all of them and double click. And then let me rescale this a little bit. And yeah, actually we can see it already right here. So there are blinks in the frontopolar channels, which is very usual. And what I would see at the same time in the gaze channels is invalid data and a straight line. And if I would go through the data, I would expect to see this more often. Yeah, and you see this is really the case. So there is this alignment. And why is this? So whenever there is no eye tracking data, Analyzer cannot do anything and cannot import any data points. So it will instead just add a straight line. So this is a linear interpolation between the last and the first data point while the eyes were closed, which is the case when people blink. And then Analyzer just inserts an invalid data marker that we can see here in red. And yeah, that this marker coincides with our blinks just shows me the data is aligned. So this is for me just a proof of principle. Okay. Then before going into the analysis, I just want to make you aware, if you right click on the channels node, there is this recording infos button here. And here you can see actually a little bit more info that has also been imported from the eye tracker. So all kinds of calibration data. So if you're interested in checking that, it's also taken over to Analyzer. And also you can, not just for the channels node, but for every kind of node, you can click on the, where do we have it? Operation infos, and then you will get again a summary of what parameters have been used. Okay, then let's now do our analysis, which will be a rather quick one. And I actually have prepared it already in a history template. So that's what we use for automation. There are 3 nodes in here. The first one is the segmentation, which will just grab out all of our E1 stimuli. Then the second one is the baseline correction to correct for offsets. And the last one is the average. And I will just now drag and drop this onto my filters node, because I will prepare this first for all the trials to create my standard ERP, which is like EEG only, so to say. If I look into the operation infos of the segmentation node, I can see again, I use the E1 stimulus and I use -50 to 400 milliseconds. So this is rather short, but long enough for our visual evoked potential. And I get 100 segments. And if I double click on this node to visualize it, I can actually see right away that I have 100 segments. The baseline correction is just making use of these first 50 milliseconds before the stimulus appears. And then we have our average. And let me just grab out 01. Hopla, it's a bit too much. And I can see very nicely here, there is a reaction to the visual stimulus. Please note that positive is plotted downwards here. So I have this P1 ERP first, and then also this N150 wave. So this looks quite nicely as we would expect it when people are attending these kind of stimuli. So let me actually just rename this node now so that I know this is EEG only. And now let's do the same for the data with eye tracking. So what I will do, I will actually just drag and drop this node again to the add channels node. And as you have seen, it will always take also what's hanging below. And now I have created the same thing on the channels node. First of all, this is still the same analysis, so 100 trials, because it's still taking all E1 stimuli in. But now I can do something very nice, because I have now these AOI markers and I can make use of them. So I now right-click here and use the Edit Parameters Reprocess option. This will just reopen the segmentation transformation, and I can now just change some parameters. And then the nice thing is it will be recalculated, and everything hanging below will also be adapting to the changed parameters. So what I can do is, first of all, nothing on the first page, but on the second page, where my 100 E1 markers are selected, I can now make use of the advanced Boolean expression. I will not go into too much details about this, but if you're interested, I invite you to go to our webinar channel and watch the ERP analysis webinar recording, because there we are explaining this in much more detail. So what I do here now is I'm writing an expression in here that will take care of evaluating the presence of the AOI markers. Yeah, so I can see my AOI markers here. And I'm using a syntax that is also explained in the manual. And let me walk you through this. So first I use the operator first, and then I need to give the type of marker, which is AOI. Then I need to give a name. And here I just use an asterisk as a placeholder because I just have one AOI marker. So if I would have more, I really should specify the name, but I have just one, so I just use the placeholder. And then I'm using a time window -10 to 10. So what is this going to do? It will take all the 100 E1 markers, but then it will check is in the vicinity of my E1 marker, namely in -10 to +10 milliseconds time range around it, a AOI type marker. And if yes, it will only select these trials. If no, it will drop it. So this is it in very brief. And let's then go to the next page where we have again the settings for the interval length, and I will just keep them. So as I said, this is now recalculating, and it will have an effect on the following nodes. So the thing we see right away is now we're having 51 segments now, yeah, because our subject kindly looked away in half of the trials. So we have a very nice balance, like 50 trials that were really attended versus 100 trials where half of them wasn't really attended. Okay, and the easiest thing I can do now is just make an overlay and look at the two ERPs in parallel. So let me just rename this average, because you can see it took over the name from my template. So I will just rename it from EEG only to EEG plus ET for eye tracking. And now I go back to my ERP that I have created with the 100 stimuli, and I drag and drop in the other node, and it is represented in red. And there we go. So what we can see is our P100 wave and our N100 wave still look very nice. They are also peaking at the same time point roughly. So we can really say, even including all trials, it's still aligned. But what we see is that if we're using only those trials with the attendance confirmed by the eye tracker, we are getting a much larger ERP. Yeah, so we have a much nicer effect here. And that's already what I wanted to show you. So I hope it was a nice, a nice example. It's very simple, as we said before, but I think it can make a nice point why the eye tracking data is actually very useful. Okay, then let's go back to the slides shortly. And actually, we are now at the end of the presentation and coming to the Q&A session, where I will hand over to Christina again. But before I do so, I wanted now to make you aware of a small poll that we want to run with you, because we are actually interested to know what's your background. Have you worked already with EEG data? EEG data only, or maybe also with eye tracking data? Or have you already worked with the combination of both? So we would really appreciate if you take the chance and vote in the poll that should be visible now for you. And with that, I would like to hand over to Christina.
Okay, everybody. Thank you so much, both of you, for your talks. I'm going to share the results of the poll now. We see here quite a nice mix, actually. So roughly half of you have only worked with EEG, 25% with both, and 28% with only eye tracking data. So it's quite nice to see such a mix here. And I think that's really nice that we were able to show both kinds of data in this webinar today. So thank you everybody who responded. A few more to ask. One would be for Laura, the first one. Can you please speak to the recording time, the average recording time for the RNet?
The average recording time for this electrode system. I think I briefly mentioned it in the presentation, so you can record with it for an hour for sure, maybe 1.5 hours. And then also depending on how warm it is, the sponges are gonna dry out slowly. So the signal quality, so the contact between electrode and scalp is gonna decrease. So if you're using the RNet, you're basically limited to say 1.5 hours before you would have to add more liquid, which you can do with a pipette if you want to. But for example, if you wanted to use the setup for a longer recording, the actiCHamp Plus runs for up to 24 hours basically. So you could also use a different electrode system, for example, the actiCHamp or another gel-based electrode system that can last for recordings of several hours if you want to.
Mm-hmm. Perfect, thank you. I have another question for Laura. Mm-hmm. Okay, we have somebody who's using E Prime. Can they still use that for eye tracking with the Spectrum?
Yes, so E Prime is another experiment control software, right? So in our setup, we have used ProLab, but of course you can also, if you already have a setup with E Prime, you can keep using that. There are E Prime extensions for both our hardware and also for Tobii screen-based eye trackers. So with these extensions, you should be able to show your task with E Prime, control your recordings with E Prime, and then the extensions also let you import important event markers from E Prime to Tobii Pro Lab for analysis. So in that case, you would need to start a different analysis project, so not a screen-based eye tracker project, but an external presenter project, and there you can import data that was recorded with E Prime.
Okay, perfect. Thank you so much. Michael, I have a question for you. How would a pipeline for fixation-related potentials be different?
Yeah, this is one of the things that Laura already mentioned in the beginning, what people with eye trackers and EEG would maybe do. It can, of course, be very different because it can be very much more complex with the paradigm. But if you just think of maybe the easiest thing you could do, similar to our example now here. So what we did now is we used the markers for the stimuli and we time locked to these stimuli. And then we just used the fixations in the AOI to cross-check for our stimuli that were attended, but we still had it stimulus locked. So for the fixation-related potentials, we would have to re-lock our trials to the fixations. As you could see in the demo, fixations were among those markers that I could just add from the Tobii Pro Lab because they were also contained in the PLOF file. So I guess it depends, and there Laura might correct me, of what you tick on or off in Pro Lab, what is taken over into Analyzer. So if I just make sure that my fixations are taken over, I could simply use those markers in Analyzer and lock my trials and segmentation to those, because I had just selected the E1 markers, but I could have just selected a marker called fixation. And of course, fixations happen all the time. So also for the same stimulus, a fixation might happen not just once, but a couple of times, because people's gaze is like wandering and refixating. So I might then actually make sure that I'm using just the very first fixation to stimulus. So you could in that sense turn the logic that I presented around, and you would then use the fixation for the stimulus for the trial locking, but then you would use the stimulus marker, so our E1 in the example, in the Boolean expression and make sure, well, just use that fixation that is the first one after the stimulus was presented. And then I'm sure that my trials are really just based on the first fixation. Yeah, so this is just one example, but basically whatever you import from markers from the eye tracker, you can make use of it in Analyzer, and markers can always be piped into the segmentation.
Perfect. Thank you so much for your very detailed answer. Laura, I have another question for you, and it's about the BrainAmp DC and if it can be used with eye tracking.
Yes, so the BrainAmp DC, just like any other amplifier, can of course be used in a stationary setup with eye tracking. Again, the main thing is that you send synchronization markers to the eye tracker and to the EEG amplifier in some way that appear at the same time. So just like for the actiCHamp Plus, we have this trigger input port at the back. For the BrainAmp, we would have, for example, we could work with a trigger box and send them to the BrainAmp via the trigger box. So basically, the main factor is you have to set up synchronization triggers in both data streams, and yeah, the rest will be done by Analyzer. And these two hardware systems don't really need to be connected in the beginning, so you could use any EEG system, any eye tracking system together.
Great. Thanks so much for that answer. I think we have probably one more question, and this one is, can the eye tracking data be used in combination with the EEG to inform the ICA analysis for blink removal?
I guess that's for me. Yeah, so that can be done basically. So normally what we would do for ICA for blink removal is using, I mean, in Analyzer we would do this. Generally for ICA you wouldn't need to really look at the blinks, right? So you could just use all the data channels that are there and could make a decomposition out of that and then find the blink signal in the EEG and take it away. However, in Analyzer, for example, and this is also the case in some other software, there are also algorithms that are specifically designed to help with the ICA removal of ocular artifacts. And there you actually enter channels that should be used for telling the algorithm where the blinks are. And if I have the blink markers already kind of from the eye tracker, I can just use them, of course. Then I wouldn't have to run a detection in Analyzer, which is something that is very easy though, but I could, of course, then use also both approaches and check the correspondence. So if I then use these blink markers from the eye tracker and I tell the algorithm where the blinks are, then it can, of course, make use of these markers to check in the ICA what is the components that is related to these blinks. So I could definitely make use of it for eye blink removal. Yeah.
Okay. Thank you so much. One more question for you. Which algorithm does this, which algorithm is used to resample both data sets?
That is a very good question, which I luckily checked earlier. It's a cubic spline interpolation.
Okay, thank you very much. So I think we are going to wrap up now. We answered a lot of questions, and if any of your questions remain unanswered, we will respond to you via email. So I would like to thank both of the speakers for their excellent talks. First of all, it was great, and all of the audience members for participating and having such a lively chat in the questions. Now that the question period is over, I'd like to take this time to share some more information with you. There was a lot of valuable material presented in today's webinar, so make sure to check out these extra resources to learn more, and take a look at the Add Channel section in BrainVision Analyzer manual, as well as Laura's article in our press release, where she walks you through a workflow, a very comprehensive workflow for your lab experiment. And I'll also point you to the introduction of the new Add Channels transform. There are also some great resources from Tobii Pro on their website. And as always, if you're interested in pricing or want to do a hands-on demo, please get in touch with your local distributor. Once again, thanks everybody for attending. We hope you enjoyed and learned a lot about our end-to-end solutions for combined EEG and eye tracking recordings. If you like this webinar, be sure to check out our next EEG and eye tracking webinars on May 10th and 11th, where we'll go over a mobile and wearable setup. Before leaving, we invite you all to participate in the exit survey before signing off. You'll also receive a follow-up email with the link to the PDF slides and a recording of the webinar. Please check our GoToStage page where you can rewatch this and past webinars, and follow us on social media so you can stay up to date with future online events we will be hosting. Thanks a lot.
Thank you. Bye.
Thank you. Bye.